So, hello all, good evening. Welcome to week six live session for the course of Introduction to Proteomics. Today we will be starting uh, with today's live session. Just a minute. So, I'll be sharing my screen and we'll start uh, talking about today what we will be covering. So, today we will be covering mostly about our some basic concepts of multi top and the hybrid configurations of the mass spectrometry and specifically of the digestion procedure like the in-shell and the install digestion. And then finally we will be starting with our uh, uh, I will be covering some hands-on on to how to interpret the data, that is how to interpret our uh, the data that we run on the multi-top or when we get the MSMS data, how we can try to identify the peptides of the protein. So from we know that from previous week uh, uh, session and of all the courses that we were studying that we get tend to identify, we tend to generate the peptides, we to convert it into ions and then we inject it into the mass spectrometer and then we just to get an idea about the M by Z and the tandem MSMS or the MSMS fragmentation pattern lead us to understand what are the fragmentation patterns. So from that fragmentation pattern, how we can identify particular peptides and protein. So that's where we would be mostly looking into in this today's session. And for before converting it to ions and injecting into it, so we had talked about like the uh, reverse phase chromatography and to elute the peptides out. But how to generate those peptides? So, how what are the methods? That is like in salt digestion or in cell digestion. Apart from that, also what is important is that how we have talked in detail about 2D gel electrophoresis. So, how we can combine 2D gel electrophoresis with MS and after doing the 2D gel electrophoresis, if we see that a particular protein is uh, expressed, protein is expressed in that particular, uh, if that protein is expressed in a particular condition only in the gel, that spot is showing to know or identify what that particular protein is or what that FYZ values can be. So then what we can do is that we can go ahead and excise that spot and then do followed by digestion, and then we can identify the peptide. Now we will be also seeing one particular hands-on with some software that is the mascot of the matrix science. So now moving on. So basic we had seen that about MALDI. So MALDI we know that it is your what was the name? So name is stands for matrix assisted laser disruption ionization. So this was one of the first soft ionization technique that was uh, discovered by Koichi Tanaka and was also awarded the Nobel Prize. So mass spectrometry uh, or the ionization procedure, which was generally used, was hard ionization technique. So this hard ionization technique that was used was not very much being able to, if we do not able to control the ionization technique or ionization thing, then what happens is that our biomolecules or biological molecules or biological molecules we are not able to uh, uh, we are not able to analyze. So when the ionization procedure could be controlled, that is the soft ionization technique, we could get it. Then what happened? So when we get it, so what we get it is that soft ionization uh, technique. So that was the one. So this using multi that it can be controlled, the ionization can be controlled, and we can analyze the protein of the peptide. So this was one of the things. So this was that for analyzing biomolecules like protein, peptides, and polymers. Now, what is this matrix preparation? So after this matrix is done, then we in the vacuum we can generate the ions that are generated can reach to the particular analyzers, and then we can identify those particular files. Right. So now matrix. What is this particular matrix, or how this matrix is prepared? What we can say is that the matrix is the small organic molecule and it absorbs the lesser energy and assists in the disruption and ionization of the sample. So this is the one very crucial is in the case of multi is that the matrix, because this is where the samples get absorbed because it is able to absorb the laser energy and assist with your disruption and ionization of the samples, and it is generally a very small organic molecule. So what are the common matrices that are used, like synapenic acid for proteins or alpha cyano for hydroxynamic acid for peptides? So these are the two common matrices used. So how it is done? So this is known as the procedure is known as sample co-crystallization, where the analyte is mixed with the matrix solution. The analyte means your particular sample, whatever you have extracted out the particular protein. So if you have extracted protein out, so then that is called the sample. So that you will be crystallizing with the matrix, and that is known as the sample co-crystallization. Now the droplet of the mixture is deposited on your target plate, and it is particularly tried to form your crystals. Then final is the laser ionization, that is laser pulse ionizes your matrix, leading to the ionization, and ions are released into the gas phase, which is injected through the vacuum, and then finally goes into the Basel analyzer or detector for further analysis. So this is the time of flight, which is in your mass spectrometer is coupled, which is generally lead to the configuration that known as your particular multi top. So this is the configuration with multi and top, which leads you to find out that the ions that are generated actually what M by Z do they belongs to, and what are the particular peptides or particular identification of that particular molecule, right? So the ions are accelerated in your electric field, and the time taken for the ions to reach the detector is measured, and it is inversely proportional to the M by Z. So that means that the larger the M by Z, so it will take the that it will get resolved on the basis of the time it reaches, time it takes to reach the detector. So detector records your time of flight producing your mass spectrum and shows the distribution of M by Z ratios of the ion. Okay. So this is the uh, particular thing which we can talk about. Like this is the target plate. So this is our particular matrix. So here is our particular analyte spot. So here comes the laser beam. It causes the disruption. So this is the H plus, that is the proton transfer happens. So desolvation and ionization, and then finally goes to the analyzer. So this is the basic principle. So this is contains your analyte plus matrix. So what is this matrix? This is small organic molecule. So this particular matrix is a small organic molecule. So where the laser beam comes, and particular ionization through via proton transfer is taking to the mass analyzer. So this is the analyte spots, the yellow ones, and this is the matrix. So this will be like synapenic acid for larger proteins. So which we all will also look in detail when we uh, go into some of those slides. You will be seeing that we are looking into it as well. Yeah, so we were talking about the 2D multi and 2D gel electrophoresis and multi-top. So the basic principle or basic idea behind here is that, so we have sample. So we have used the protein features. So if we have used protein features, where particularly we have tried to use this particular like PI and like CI and molecular weight, and we divide it. So for example, if you are looking, right, so this particular spot has certain PI and certain molecules. So if you are interested, so for example, this protein is highly expressed here. So overexpression means seen in only one condition. Overexpression. Overexpression is seen in only. So then you are doing sample preparation, and after sample preparation, you are, for example, after you have prepared the sample, so you are taking the sample plus matrix, spotting in the plate. So sample plus matrix, if I see, go ahead and spot in the plate, and then you are injecting into the vacuum for getting the MSMS spectrum, right? So we are getting the MSMS spectrum. So after we get this particular spectrum, then you can identify this particular peptide. So we would be seeing that how we identify some of the hands-on session, which we will be seeing just now with one data file. We will be trying to get this particular thing going. Okay. So now is why. So how it's done. So here it is very important to see this particular cell. I think all of you can see. So this is the particular cell band. If you want to see, this is basically the general procedure. Okay, so this is the general procedure where you take the gel piece out, put it in the level, and then finally you do. So this is for the gel register, right? So now if we get for this uh, particular thing, that is if we get for the gel register, that is if you do this, so we can get it. So now this is a simple workflow that we see like the protein and complexes. So we are doing solubilization. This is a 1D page, which can be. So this is, as I mentioned, that if the general procedure are not restricted to 2D, but of course we can use it in for any kind of gel-based procedure. So if we have the STS page, then we have 2D, that is IF STS page, or native is take this particular band at this particular spot or this particular spot. So we are going to do initialization. Then you can do multi SIMS or use IMSMS. So this is so if we are having multi MS or just SI MS, so the basic difference that if for any particular thing, if we get only for MS, so we can have PMF. So what is PMF? If this particular question in this particular thing, so PMF stands for, if I say PMF corresponds to peptide mass fingerprint. Right? So PMF stands for peptide uh, fingerprint. So why it is there? So because of a single MS file, what we will be getting that only the overall molecular weight of protein. So what we're getting is that overall of the particular protein. Now for the, if we do for MSMS, so what we can get it is that the particular sequence of peptide. So this is the difference. So this is where uh, mass of the protein, that is for the peptide mass fingerprint, whereas MSMS we are getting only the, we can reduce the sequence of the peptide as well. So that's why it is written either through multi or ESI. We can perform multi MS or ESI MS, or multi or ESI MSMS as well. So this is the basic difference, and this is a general workflow how we can do. That is, we can take it to get it to 1D page or SKS page, and to perform the 2D page, that is IEF SKS page or 50 SKS page, and to followed by in-gel digestion or proteolysis, and then get the acquired the data to multi or SIMS or multi SI SMS, and finally we can interpret the data to PMF or de novo sequence. The general procedure. Now I have told the difference between peptide mass fingerprint and or those particular database databases where we can identify. And I think you have gone through the videos to see that where you have got about a particular what I can say you that uh, where we need to couple two top mass analyzers or two quadruple and top mass analyzer together, to that you can, so that particular thing you can get it, that is the top or top quadruple followed by top or two top, to get the particular what it is in the sequence of the peptide. So this is overall. Now how we are going to do it, that for the in-cell digestion, if we see, so basically how the things work is that we get the lysate. So from the samples, biospecimens, we were preparing the lysate, that is lysis, fractionation, diffusion, enrichment, dialysis, etc., etc. So all these belongs to the lysate preparation from the sample biospecimens to then finally we are going for in solution digestion. So if this is the place where we have our samples. So for example, if we are taking, so I'll just give you an example from cells or tissues. So how we were going to do it. So you have what with, so if you have to wash it with particular uh, 1x pps, and after you have washed it, you are going to add, you are going to add particular lysis buffer, and then what you are going to do after you have added the lysis buffer, you have to go for the tonification or homogenization with the to form particular solution, right? To form a 10-second, yeah, to form particular of the samples from the cell lysis. So what we are going to do is that we are going to prepare the samples. After we are getting, that is after washing with water, prepare the lysis. So why this step is required? The cell, all these things is broken out. This is an intact cell, and this will be this is an intact cell, and this is the broken cells. So this is the, so for instance here all the proteins are there. So these proteins will be coming out. So for that in the form of solution. So proteins are in the solution. Once you have the proteins in the particular solution, so then what you are going to do is that uh, this you will be either taking. So now this is the step we have of our protein, right? So this is the step where we have the particular. Now if we have our particular proteins here, so now what it is happening is that either there are two ways through which we can proceed. We can go for one-dimensional electrophoresis or two-dimensional electrophoresis. Another through is that through in solution digestion for proteomics. Finally, our goal is to run in mass spectrometry to get proteomics experiment performed to identify all the particular proteins and finally getting data uh, out of it, right? So this is what we are going to do finally. So now what is going to happen is that we are going to proceed with either of the steps. That is either to, yeah, so either through this route or this route. So now, for instance, if we go through this route, so we will prepare the chain and run our gel here, which shows our protein batch. Now we can exercise one particular bat and take into the tubes and perform the solution. Now, this is the state where you can see that both our protein, either our protein or the gel with the protein, gel band with the protein is in our particular solution. And then we will be proceeding with the same step of digestion. So that's why this is given in the same uh, column, if you see, because it is depicting that it is at the same step where you have the protein either in the solution or in the chain, but in the solution. So now the steps are the simple production and digestion, and then followed by peptide enrichment or cleanup, and then getting the peptides. So now what we are going to do is that we will be following this particular uh, step, and we will see that what this is meaning of particular reduction, appellation, and digestion, and also we'll be saying that how what is the step for peptide cleanup or that is salting actually refers to what and what does it mean, right? So this is what we are going to do it. So now before uh, going into this part, so we have so because in the Maldi of also you see that uh, it's better to particularly regenerate out, to particularly clean up the samples so that we can have an improvement of particular protein or peptide. So this is why the concept of peptide desalting, or which is known as your uh, peptide desalting. So now the same concept, the desalting of the particular peptide, right? So the peptide desalting is follows also follows the same principle, same principle of what we get in for particular. What I can say is that what the principle through which it is run in the column, and we get the particular solution out, which we had talked about and which we have also depicted the uh, gradient. But what happens is that okay, so what we are saying that the principle of which we are diluting, through which we dilute peptides out for gradient analysis. Similarly, we are getting this is the principle we use for desorting. So why we use this reverse chromatography? So if you just think of the chemistry that the peptides are hydrophobic in nature, right? So peptides are hydrophobic in nature. So how we are doing is that we are using one created stage tips. So what does it do is that it contains uh, it will let the particular peptides will get bind to it. The peptides generally get bind to it. So how we do it? So first of all, the stage tips or these particular tips. So it happens within a particular tip. So in the tip we will be adding the creatine material. So how we are going to do is that we are activate the creatine tips like by addition of ethanol and addition of ACN will be activating it. And once we activated it, then what we are going to do is that we will be doing the digested peptides within the tubes, right? This we will be passing for in loops, like in iterative steps, for 5 to 6 times. So why we are passing it for 5 to 6 times? So that our particular digested peptides get bind to particular to the to bind to testic, right? So this our particular digested peptides gets bind to the stitch tips, and while the samples is uh, while the remaining salts, so whatever remaining salt are discarded out. Remaining salt discarded out. So once it is done, so what next is that? Next, you need to wash this column so that any discard is go through it. Wash the column. Then finally, what is needed or what it is generally done is that you will be taking this particular tube, you will be adding your particular tip. So if you add this particular tip, then what will happen is that you will be using different gradients of organic solvent like ACN. So why gradient? So this will create the hydrophobicity. So with increased hydrophobicity rather with than the particular stage tip, but it is there. So where it is bind to it. So if you increase the hydrophobicity, so the peptides will get dissolved into the particular uh, hydrophobic uh, gradient. And then what it will happen is that the peptides will keep on diluting out, and the peptides will keep on diluting out until you get the peptides. So this is what we generally tend to. So that's why we increase the gradient as we do for our particular during the illusion of the peptides for reaching to the mass spectrometry. Similarly, is that we speed up the peptides using the reverse test chromatography where we increase the gradient of the peptides and do it. Okay. So so I think I'll repeat it just once more that the resulting peptide resulting involves to remove the salts and uh, other fractions of waste that stays within this particular peptide. So it is done through the process known as you can tell it's reverse phase chromatography. So how it is done is that there is a hydrophobic material which we use, which is basically a C18 tip, which has because of the hydrophobic property when we flow through the sample with the peptide. So what happens is that peptides gets attached to the particular tip and the samples are discarded out. And then what we do is that uh, we increase the gradient, the hydrophobicity, and in this way we elute the peptides, and in this way our whatever salt and other things are there that cleans up our sample for particularly for our particular analysis, further analysis. So this is how a basic 196 well plate of uh, uh, multi-plate looks like. So here you can see that the samples have been mixed with the matrix and it is very well mixed and then spotted out when it is dried, and then finally it is the laser is ejected and it will eject the charged molecules and it will flow through and reach the rejector to get the ideal MYC values. So this is quite important and uh, which is quite important for getting the particular uh, data. So you can see this is a simple basic plate and you will spot these particulars. So here what happens is that when you inject, so for example, this is the compartment, this is your plate, right? So this is your plates. So what is there is that your injection in this chamber, when you injecting it, then when you in the software, you will see, so you will detect the spots where you need to add the lasers, add the energy lasers, and what it will do is that it will just eject desorption will occur and it will let the particular ions to reach to the detector to get analyzed. So, but what happens is that we will, so what we will be getting is this particular protonated ions, that is N plus H plus ions, and that we can identify that what is a peptide mass fingerprint or two. And if we use it in a top configuration, so then we can get it that particular thickness of the peptide. Yeah, so this is how particular things is happening, where we have the matrix, there we'll be getting analyte, the sample. There are analyte ions and the samples, analyte matrix mixture. So we will have the matrix ion and the analyte ions. Then we will be having the laser beam sample electrode. So we'll be just taking this particular analyte ions, structure electrodes, and then getting to the mass gate, electron linear shifter, and finally you are getting this process. So this is the schematic of how your particular ions are generated and goes and finally reaches the detector. So this is what we can say that if we are if you are told to design if you are asked to design a simple so if we are going to decide this simple Proteomics experiment, so what you can do, so just remember from the previous slide that I had shown. So you will, so this is basically what uh, workflow you can choose to deduce your particular hypothesis or experiment or result that what you'll be doing. So first is that prepare the lysate, we either install or run in the jail, get the digestion, and then finally, so here you will, so for example, if we take for the engine digestion of the 2D gel and multi try to mix it, so you will be getting those impressed size shell, then finally getting the protein digested with the trypsin, and then it will be cleaning up with the creatine tape tips samples deposited on the plate. So if you are doing for multi, it will be mixing with the matrix or the light, and then it will be performing multi-top or data application. So in this, you can understand from the name itself, multi, which can be talking about that why matrix is important because we could control the ionization procedure, which was not possible before with the hard ionization techniques to uh, analyze the biomolecules. So what we are going to do is that we were getting this particular ions, that is a particular matrix and particular analytes. So what we are getting these particular things. So we are not so you can understand that the matrix plays a very important role in this. So what is the role of the matrix? So matrix first role is that it absorbs the laser energy and it helps in sample absorption and ionization. So it helps in sample to help in the sample. Uh, there helps in the there helps in the sample absorption and ionization. So this is the one thing. Second is protects the analyte from the direct laser damage. So there are two things. One is that to it absorb the laser energy and that aids in the sample absorption and ionization. And another thing is to protect the analyte from the direct laser damage. So the analyte is your here analyte refers to your particular sample. so that it gets protected from a direct laser damage. So these are the both roles of the matrix in the broad sense, that one is absorbs the laser energy and it's in some absorption and ionization, whereas other thing is that to protect the analyte from the laser damage. So what are the criteria for matrix selection? So first of all, UV absorption. So must strongly absorb the laser wavelength, which will sorry, which is generally the UV. So that that thing should be uh, done. Then the criteria is that the ionization efficiency should promote efficient ionization of the analyte. So it should have a great efficiency for uh, having the ionization of the analyte. It should not lead to have this. If it is not having efficient ionization, then it won't be of any use. So that's what very important thing. Chemical compatibility, that it should not react or degrade with the analyte. So obvious is that when we are mixing our sample with this with any particular analyte, then definitely should not happen that what we are getting is that yeah uh, so that is degrading our particular analyte. So first of all, that it should absorb the laser energy, sample disruption, protect the analyte. Next is that it can absorb the UV light strongly and laser wavelength, specifically the UV light ionization efficiency should be efficient from efficient ionization. Chemical compatibility should be that must not react or degrade with the analyte. And then what we will be doing is that volatility, that it should evaporate efficiently under laser disruption conditions, and commonly used matrices are like alpha cyanophosphate acid, that is ideal. So these are some of the commonly used matrices. So one like which does not, which promotes efficient ionization, or which is quite compatible or it's not degrade the analyte, and also evaporate efficiently under the different conditions. So these are very much used for peptides or small proteins that are less than 5,000 daltons, and whereas synaptic acid is used for proteins, larger proteins, for higher than 5,000 daltons. So this is one thing. Second thing is that suitable for matrix analyte focal stabilization for oligosaccharides and glycoproteins is that a fluid matrix forms homogeneous crystals. There should be reproducibility and consistency in ionization. And these are some of those application specific. Uh, we can have application specific matrices as well, like picolinic acid for nucleotides and other things. So this is the overall. So there will be different types of matrices. The primary role is to absorb it and to provide efficient ionization and without degradation. So it can be used, but these are the commonly used for peptides and the proteins and what can be taken forward. So now we had talked about multi. So I was talking about those that if you have only MS and MSMS, so MS will give us that PMF or peptide mass fingerprint, which we have talked about. But when we want to get reduce the particular peptide, so we need to have a hybrid configuration of the mass spectrometer. Where multi-TOF we had talked about, but multi-TOF is another method where you will be getting that there is two hybrid. So means there are two configurations of the mass analyzers, and obviously in the previous week we had discussed in detail about the mass analyzers. We had talked about those particular things. So here you can see that this is our particular multi-slate. So here, if you get the lizard detected, it will go here, then again go through top and then finally reject it. So how does it happen, right? So how does it happen? What is the basic principle? So we have seen the diagram. What is basically the principle of multi top of MS? So that's what we are now going to see. You can see this is a target. So this is the top one and this is the top two. So this is the two top time of flight mass analyzer that are configured both in a very uh, sequentially to mass analyzers are attached. So here how it happens is that ions are there, targets where ions are dissolved and ionized from the microtiter plate and accelerated into your first top analyzer and mistrustable and collision induced fragments continue to travel the intact molecular ions they are derived from. This means that this two goes together in space because you can see here, you can see since if you are not giving the enough space for them to separate it out, then what happens is that so ions are dissolved and then ionized from those microreactor plate and accelerated into your particular place first top analyzer, then having metastable and collision induced fragments continue travel with the velocity of the intact molecular ions they are derived from, and where it happens that they stay close together in the space, then top one ends with the selector. So already analyze of the so what does it means the selector? So from the name that you can deduce what derive as from the name you can deduce and derive is that only analyze of the selected mass and the derived fragments. The selected mass and the derived fragments can pass to the ion source of the second top parent. So this all other molecular ions are checked. So this is the term of the selector. So in the first stop, if you are not giving enough space, so they will remain quite close to each other. But what you are doing is that you are selecting those particular particular masses, so only those ions that can pass to the second top uh, are passes from through the selector. Others are not allowed to pass. So all other molecular ions, whatever are there, are rejected. In the ion source of top two, the selected parented fragment ions are accelerated. As subsequently, these analyte ions separated in the conventional TOF2 analyzer. This allows the mass analysis of the fragments by measuring their arrival time on the detector. Okay, so this is TOF stands for your time of flight. So this is how it goes. So first of all, it is going. Particular ions are being separated in closed space, and there's a selector. Then you are going to select only those specific ions. So you can see that this one ions are only selected out and not this one. And then what happens is that you are separating it out. The second top spectrometer, and this is how it is going, right? So that is it is only the selector plays some role where you are selecting on those particular devices. So if we talk about in the graphical sense, so from the mass spectrum, if you see, so this is your embodies, if you say abundance, it is relative abundance. Okay. Right. So now what we are saying is that we are getting this particular spectrum, right? In the, for example, what I can say is that if I see, this is the particular at MS1 level, right? So if it is at MS1 level, now what we are going to say is that if we are going to take it, and now if we select, your selector is selecting this M by C, okay? Selector is selecting this particular M by Z, and then it is getting this, gets your particular fragmentation, and then finally you get the spectra for this particular selected edge. This is how it is going on at the MS2 level. Okay, so this will give the PMA, this will reduce the sequence of the peptide. So this is how it is done. Okay, so now if we move on. So if we want to calculate the isotopic mass for a particular molecule, then how it is done. So based on the, if we talk about that, based on our particular, mass spectrum below given, then how much we are can isotope abundant, right? The average top will be sigma into isotopes into absolute percentage of abundance. So here, for instance, it is given, so 78.92 M by C, so 50.69, so 78.92 50.69 by 100, that is percentage, and 80.92 into 49 by 100, so average mass will be 79.91. So this is what we are getting. So if we have an isotopic mass and of all the isotopes and their relative abundance or presence of the abundance of those particular isotopes, what we will be getting is that particular mass of that particular thing. So we will get like 78.92 into 50.969% by 1 and 80.92 into 49%. So that's what we are getting at 79.91 is the which we are getting. Now, if we talk about basic in the about the steps for if we say in the angel digestion, if we talk about so now how we are calculating to know there are steps we are saying easily that we have run in jail, we are taking part in the tube. We are taking a tube, but this and we are going for digestion. But that's not the actual step. The actual step involves other things like the chain will contain the stain of Kumasi, right? We are having the Kumasi strains. For this, we need to remove the excess Kumasi blue dye from gel bands. So that's how it's done. So we want to massive blue like some more hydrophobic things. So we will with the 50% HCl and 25 minutes of bicarbonate, then should till the time actual basis should appear nearly colorless, indicating effective staining. Then how we should do the dehydration is that prepare the gel pieces for subsequent steps by removing water, HCl, opaque and shrink. Gel is ready for rehydration with agents in later steps. Once it is done, so we will be using induction that is through PTT or IA. So this induction means that this is this is disulfide bonds to break into SH, right? So this is use dithiotretol component at 37 degrees C or 30 to 60 minutes. And then is that alkylation. So once it is done, this is reversible and it can again bond to disulfide bond. But what happens is that we can use iodoacetamide. So this will form a CH3, is CH3. So this will alkalite and this will help doesn't reform of a particular disulfide bond that we can finally exposing it so that it becomes easier and we can go for the final efficient digestion by using different enzymes. So we should remove excess reagents, prepare the pill for digestion, and we should treat it again with designated. So now the digestion means that we need to put your critically brief proteins into peptides for mass spectrometry analysis. So for that, what we should do is that we should add trypsin or other specific proteases. So why these specific proteases that right? So why these particular trypsin or other specific uh, proteases? So the reason for this particular thing or trypsin or other particular proteases is that we tend is that we what I can say is that so this has a specific what Okay, so this is the trypsin that we are using the specific protease. So why we are using the specific protease? So if you have the specific protease, so we know that where means I mean to say that in which particular position it lives, right? In which particular position it lives or it will live. So that is what very, very important, right? So this is what very, very important. So which position, which particular position it lives. So that's why because it will know that which particular position it lives. So then what we can do is that we can uh, match with the theoretical mass spectra for this particular proteins and then finally match with the database for enzyme symptoms. So we'll rehydrate with the enzyme solution, that is trypsin or other proteases, specific proteases, and incubate overnight at 37 degrees C, and then peptides are released into the solution for subsequent analysis. So how this, what are the specificity of some common endopeptidases? So these are all endo endopeptidases, like C-terminal site of positively charged residues, arginine and lysine, but RN should not be proved so that if there is an immediate proline that will enter it and will not get the desired result. Or which we are talking about is the chymotrypsin. So this chymotrypsin is what? So which is bulky hydrophobic amino acid residues, that is phenylalanine, is phenylalanine, tryptophan, and tyrosine. So again, here also this must not be proline. Then last is on neutral residues, on proline, pepsin, and thermolysin. So enzymes vary in specificity pocket. So what happens is that vary in amino acids in pocket, which controls your chemical environment. So controls pocket dimension. So if we say glycine residues make deep pocket, pocket fits around aromatic ring. How it does is that if we go to trypsin, we will do pocket the same under the same dimensions chymotrypsin, but different sizes. Aspartate is there, which readily binds lysine or arginine. Lactase, non-polar side chains chymotrypsin. So this is how we can go and try to find out the specificity of the enzymes and how it generally get helps in binding this particular enzyme and this protein. Okay, so now we will be doing this using our particular software, that is a spot or matter science. So we will see how we can access, identify peptides or anything. We are going to identify the peptides, right? So this so in a minute I will share the screen and start. Yes, in a minute I will start sharing it. Just a minute. Right? So we are going to so what we are going to do? Yeah, so yes, so this is a web server, a simple to use tool where we can use this. And also I had given you right. So this is particular data set. So now what we need to do is that you can see here. So this is the matrix size for the matrices. So this is software which we can use to identify the peptides of particularly the protein through the spectrum that we have generated using the software. We can identify it, right? So what we are going to do is that to access the mass server. So here there are two options, right? You can see. So I just share this link as well. And so here we can see, so you can see the term is written peptide mass fingerprint, you have written MSMS answers. So both the things are possible. So peptide mass fix is that which contains of the protein by a specific enzyme such as trypsin when we are doing only one muscle level, that is coffee for instance if we are doing. So in that polytom what will have just it will have one particular enzyme for this particular peptide that it will be hitting those particular data, which is of not other things. But whereas this MSMSI search, which will be on or MSMS data that is for the tandem fragmentation pattern, which should match this particular peptides, right? So this is quite uh, important. Okay. So now we will be doing so for instance we go and do one MSMS science search. Let us do it. So in the next week, if there is another scope, I'll again do it, show it and ensure that how we can So that does not have this issue. So what we're going to do is that we will going to have mascot, SMS, and search. So we can use the search title. So this is the search will be that from section Proteomics. So you need to create your account ID if you want to do it. So since I have already an account, so it doesn't require it is automatically taken the username and the email ID. So what you can do, introduction to procurement search, uh, limitation, query search, right? So now we have the databases. So databases, one is a contaminant, one contaminant would be coming from like the trypsin, from which organism it is being done. So it is not from the human trypsin. Another thing is that uh, abundant proteins that stays in the environment and it can contaminate during the fermentation. So these two uh, comprises of the databases for contaminants. So this we need to already it has been added. Now the thing is that you can see that if you know a particular species that you are looking at, or that particular species, that particular protein is made of, then it's fully very easy for you to get particular organism like X levis, C. elegans, or sativa, or human express sequence tags, or particular libraries if you have. But what we are going to do is that we are going to add, so for all the proteins, for the comprehensive list of the proteins, what we are going to do is that we are going to get it like this plot, right? So this is the amino acid database that we are adding. This plot. Now again, there is another option for us to add taxonomy. If we want to add, let's see this Archaebacterium, carriers, alveolata, plasmodium. It has other things, or if we are not sure of which species it belongs to, and if we want to check that whether it belongs to all the particular any particular organism or not, and then what we can do is that we can click on all entries so that it can spawn up all the entries or all the possible organisms. We are not sure which one which organism it is, so we won't take the risk and we will be trying to search access all the all entries of whatever is present in the Swiss plot database. So we are adding taxonomy as all species. Then the enzymes is the we can add like these are the all those proteases which can be used, or it can be used in a combination like you can see that lyc plus SPN, CNBER plus trypsin, chemotrypsin, trypsin P, trypsin A, etc. etc. So if we are using particular enzyme, so we are using trypsin. So trypsin T is that which does not depend on whether lysine or arginine will be there if proline is cut or not. So it will be all uh, equal to it. So this is the trypsin. So now what are the bis cleavages? So if I just talk about a particular thing that is so I just open it. So if I talk about cleavages, uh, one second. Yeah, so set. So if I talk about mixed cleavages, so what is the meaning of the term mixed cleavage, right? So here it is lysine, X, Y, say A, B, C, or J, right? So this is what, for instance, suppose of the peptide is there, and before that also there is one lies, there is one. Now, if it is that here it has cut, here it has cut, but here it could not cut because trypsin has also certain its certain efficiency. It is not truly 100% efficient as of such that it will cut each uh, right. So you have uh, missed limit, right? So you cannot so that trypsin does not is not able to get cut at this position or this position. It fails. So how many these cleavages we can allow is that one of many misses that we can allow is that what we talk about is that best cleavages. Ideally, it should be zero, but in all practical sense, what we do is that we can say is that 1 to 2 we can allow best cleavages, and obviously it will have an effect. So can see by going by reducing the number of cleavages or increasing the number of cleavages, what is the number of hits that you are getting. But definitely for acceptable range is that two plus privileges are quite acceptable in the community. So the quantitation, if you have done certain quantitation, I think it will be done covered in the upcoming things. So which we'll be doing. So there is no fairly good. Second, so we are not going into the details. So this carbamidomethylation is required because uh, we have added a particular that is we have done alkylation, so which leads to the addition of the alkylated things. So what we are getting, so we are adding the particular fixed uh, modification as carbamidomethylation because this is always required uh, in the particular sample. So it's whenever we do the sample preparation, what happens is that we are adding it. So that's what we get in the fixed modification. In the variable modification, we can add. So if we want to check phosphorylation and all, those are not fixed. Rather, it depends on the status of those particular protein. So here we can use like oxidation of methionine. So if there's again the class that we get in the week to show, we can discuss it in more detail. I think it will be coming that regarding the each samples, type and the qualifications and how we can use it. Then we have peptide tolerance. So this we are keeping it default as for now. We can choose our target files. So that's we are using some MGF file, that is MGF stands for mass generated file. Okay. So instrument we are keeping default, it will take it. So you can see all these instrument types. So tab file is mass generic or MCML files that it can take. You cannot take other files. The target FDR we are keeping at 1% FDR, which is acceptable range. So we are not going into the decay of the decoy search and all. So there is another thing of the decoisers, which we would talk if we get some time in the our next week. So we are keeping the search running, right? So the search is been running. Yeah, so now you can see that we have got the results. So you can do the PMF, that is the peptide mass spectrometry as well. So I just showed it for the MSMS science subject that because for data the MSMS information. So this will give you the search parameters. This will give you the score distribution, that is the number of matches and the score. So for how many score, the number of matches are how much. The protein family sequences above homology. So here you can see that this is the protein that we have got. So this is the size pyridoxine S1 of Arabidopsis thaliana, which we are getting identified. So if we go and click on here, yeah, so you can see this is the complete sequence of a particular protein that is getting the maximum heat, among which we have the sequence coverage of 41%. So we are having the heat or coverage of this particular protein. So we are getting 41 percent heat of this particular protein. Now what we are getting is that we are getting the suspected, the score, and this particular peptide. So now if you open it, yeah, so these are the ions or these are the spectra that you get it, that particular peptide. And how you are going to get it? So from the set of Y ions and P ions. I don't know if it is the scope is there to state for your particular Y ions and P ions. So basically this Y ions different from the C terminal. So if I have spoken about like the if you go and read the paper for ABC and XYZ of peptide sequence or protein sequence. So there you can go and understand that why ions are generated fragmentation of C terminals, where the ions are generated from the N terminus ions. So we have this particular ions. So y2 y minus y7, y7 6, or y6 minus y5, or y5 minus y4, or y4 minus y3. So these are the ions and those particular ions, right? So this is how you will be introducing the sequence of the peptide. So the basic uh, prerequisite here is that to reduce the particular sequence of the peptides, we need to have the particular MSMS information. Then only we can generate the YIS. So here you can see that what YIS have been identified. So from here we have got an idea that this can be this particular peptide, and this is the full coverage within our particular protein. 41% of the peptides would be matched and identified with the score with a certain threshold, which let us to understand that okay, so this is can be our uh, particular what I can say is that particular protein. So this again you can query here. So you can see this is the number of spectrum ions that have been identified. You can see this is the ion. Sorry, I'll just do it again. Right. So we go down. So these are the Y ions and these are the P ions that have been identified. You can see these are the Y ions and these are the particular P ions that have been identified. So this is how we can do that. And I'll show you this particular thing. Now there are other things. Sorry. So target, if this is the second protein that got hit, and these are the third protein. So now you have to decide on which particular protein on the basis of your certain score and the confidence you will be. So you can see that this coverage percent is 2%. So this can be at this particular peptide is a shared peptide between those two proteins, and that's why it just got matched. So that we will not take. So here you can see that trypsin quercetum side of KR, that's lysine and arginine, until it is next residue of P, and calculated PI and those things are to provide. So this you can see that it should not be considered because this can be just shared with the sequence, and that's why it is showing that it is belongs to this particular protein. But that with high confidence and high coverage, we will be taking for the peptide protein that is having the 41% coverage as such to take it forward. So that's why it becomes quite important for us to understand that which particular protein we are to take. Yeah. So this we can, I guess we can end our thing particularly here. And of course, if you have further query, you can ask. Otherwise, we can meet in the next week and we can do. I think most of it will be the contributing strategies and the independent time. So we'll talk more about this particular software and others as well. So, and as we had stated, that whatever we will get time, we will be doing some activities for major apart from regular. And so, thank you all for joining.